So before we begin part 2 of the State Biodiversity Lab, let's go over the materials. So you'll notice you have goggles for every member of your group. You should be wearing these anytime that you're working with the chemicals in the test tubes. Also, you notice that you have a test tube rack and you should have 4 test tubes with different liquids in them, and they're labeled B, C, X, Y, and Z to represent each of the species with plant extract in the bottom. In each of those, you also have a dropper in order to get the liquid out. You should have a cup with a very small amount of water. If the last class before you didn't fill up the water again, go ahead and just fill it just above the mark on the bottom of the cup. You also have a Dixie cup that says enzyme powder that you'll use for the enzyme test. You'll have a tray with different wells. You can see there's little circles in there to put chemicals in, and they're labeled B, C, X, Y, and Z to make sure that we know which one is which. You see that you have a small piece of paper. This is for paper chromatography, also listing all of the species of the plants. And then you have 2 sheets. One is for task 5. Each person in your group will be receiving one of those. And the other is a long strip with DNA bases listed on it for each of the different species. So you'll need, everyone in the group will have one of those per person. Everything else you're going to be sharing. You also should see 2 of these genetic code charts at each station. So before we begin, let's put our goggles on. And we're going to start with the paper chromatography test. So this is for task 2. And if you look at your lab sheet, we're going to be recording evidence for task 2 here. And we're going to be writing down colors that we see in the paper chromatography. So for the setup, what you're going to do is take this piece of paper and take a drop from each of these test tubes. And you're just going to touch the surface, okay? It might spread out just a little bit, but try not to squeeze. Try to just touch the surface of the paper in the spot that matches the letter. Okay, so touch. And then we're going to do that for all 4. After you've done that, what we need to do is take the piece of paper, and we're actually going to put that into the water. So I'm going to put this to the side. You're going to get your cup with the small amount of water, and you're going to hold your paper just so it just touches the water in the bottom. And you're going to wait until the colors stop changing, and you're going to record your results of what colors you see for each of the different species. When you're done, you can throw that out and just rinse the cup back out and put the water in just like you did before. Task 3 is the test for enzyme. Again, you're going to be writing in here, and you're just going to say yes or no whether the substance bubbles or not. So for task 3, you're going to need your tray with the wells, and you're going to need your enzyme powder, and you're also going to need your test tubes. So for task 3, you will need to take, and you'll see there's a little straw, and there's an edge to it. So you're going to take that and just scoop out a little bit, tiny little scoop of the powder, and put 1 scoop in each of the different wells here. So B, C, X, Y, and Z, just put 1 scoop in each. And then what you're going to do is you'll take 1 drop, all you need is a drop, and you'll drop that right on top of whichever letter you're looking at. And if it bubbles, you write yes. If it doesn't bubble, you write no. When you're done with this test, you'll rinse this out and then return it to the tray just like it was. Task number 4 is for gel electrophoresis. That's the little sheet. And everybody in your group is going to do this. You'll notice that I did the first one for you. So Botanicurus, what we're going to do is essentially cut the DNA at certain points. So every time you see the letter C, C, G, G, you're going to cut the DNA. And you'll see that I did that on the Botanicurus line. So if you look really closely, there's a CCGG right here. And I drew a little line. So we don't actually have to cut it. And then again, you see one here. Again, CCGG here. And that's all of them for that line. After I've done that, I count up the number of bases. So here there's 1, 2, 3, 4, 5. So I wrote a little 5. I counted these. There's 12 in between my lines. There's 11 between these lines. and then 9 at the very end. So then I just reorganize my numbers in order, 5, 9, 11, and 12. Okay, those are the size of the pieces that I just broke my Botanicurus DNA into. So then what I do is on my sheet for task 4, I'm going to write 5, 9, 11, 12 in this box for Botanicurus. And everybody can do that actually now because I've done that one for you. So for species X, Y, and Z, you'll be cutting the DNA or drawing the line between anywhere that says CCGG. When you're done with that, you can actually throw this piece away because all of your evidence is now on your lab sheet. The very last task is task 5, in which you will need this half sheet of paper. And you're doing the same thing that we've done in class. You're taking a line of DNA, you're turning it into RNA, and then finding the amino acids using the chart. So I would imagine that at least one person at your table knows what's going on with this. So if you need help, ask each other first, and then ask me if you still haven't figured it out. Once you are done filling this sheet out, you're going to go to this paper, and in your data table for task 5, you'll notice there's an X for Botanicurus because it says differences in amino acid sequence. Record which, if any, amino acids are different than Botanicurus. So maybe species X, the 3rd and 4th amino acids are different. So you would write whatever those differences are and then do that for each one, okay? After you're done, make sure you've cleaned up all your materials. Again, you can throw out the extra sheets of paper because you have all your data in your data table here. And then you can clean up your materials. Leave all the liquids in the test tubes because other classes will reuse that. You're going to throw away any trash like your paper chromatography. Make sure you've rinsed out your tray and added clean water to your cup.